metabolism is a process in your body where many enzymes work together. It refers to all the activities by which the cells require and use energy. Metabolism often occurs in this big series of reactions made possible by enzymes. Here is an example of how metabolism happens in your body. Don't worry, you won't have to know this for the exam. But what I want you to see is each one of these numbered bubbles, all 10 of them, a different enzyme comes in and makes this reaction happen. Controlling metabolism. There are many ways that a metabolic pathway can be shut off or turned on. Remember, a metabolic pathway is how your body makes something, or breaks it, that's backwards. It's how your body breaks down something. So one metabolic pathway could be how your body breaks down your sugar. Some enzymes have an allosteric site, a place where a molecule other than the substrate can bind. Molecules that bind at an allosteric site can turn the enzyme on. That was kind of confusing, so I'll give you an example. This green thing that looks like a bowling ball is an enzyme. Right now, the enzyme's active site is this circular hole. The substrate that the enzyme's supposed to bind to is this square box. Obviously, the substrate can't bind right now, so the enzyme's turned off. This enzyme has an allosteric binding site. When the allosteric activator, which is another molecule, binds to the active site, the enzyme gets switched on. Take a look down at the bottom of the page. You can see now the allosteric activator, which looked like a lucky golden horseshoe, has bound to your enzyme. Now the enzyme has an active site of the right shape that the substrate can bind. Now it can do its reaction, and the enzyme pathway is switched on. In this case, the allosteric inhibitor turns the enzyme pathway off. So an allosteric activator activates it and turns it on. An inhibitor inhibits it and turns it off. In this case, when the allosteric inhibitor is not bound, when the allosteric binding site is vacant, the active site can bind the substrate and the reaction can happen. When the allosteric inhibitor binds, the active site changes and the substrate can no longer bind, so it's shut off. These mechanisms for turning on and off enzyme pathways make sense. I mean, you wouldn't leave your heater on in the dorm when you're out and about doing things. I mean, there's no sense in heating an empty room. There's no point in your body making something or breaking something down that your body doesn't need. Sometimes the end product of a metabolic pathway can act as the inhibitor for the enzyme involved in the first step of the metabolic pathway. So this is called feedback inhibition. So whatever your body makes at the end of the metabolic pathway shuts off the metabolic pathway. This makes sense. When the cell has enough product, when the cell has made enough of whatever it's making, it shuts down the pathway to save energy. That way you don't have this big backlog of product that you don't need. Here's an example. The substrate is the kind of teal box thing here, and you can see there's a series of 5 enzymes that do 5 different reactions to make the box change color until it's a red box. The red box is the end product. When you get enough end product, this end product actually goes over and inactivates the first enzyme. It works as an allosteric inhibitor. That way, once you have enough product, the enzyme automatically shuts off. Redox reactions. Have you ever tried to set sugar on fire? It works really well if you put the sugar in a chamber that's just filled with oxygen. Not regular, just pure oxygen. When you put a little spark in there, it goes boom, and it explodes, and there's fire, and it's very exciting. The glucose and the oxygen get made straight into carbon dioxide and water, and all the energy goes out fast. So releasing the energy from atomic bonds all at once make it go boom. Well, this process that we've shown in the picture, right, is the same process where you break down glucose in your body cells and you make carbon dioxide and water. But your body uses this energy. Well, there's obviously not little explosions going on in your body. What's happening is something called redox or oxidation reduction reactions. These break down the molecules slowly. It involves the transfer of electrons. It's a way of harnessing this process, so instead of it going boom and releasing it all at once, it releases it nice and slow so your body can use it. An example of how redox reactions work is the electron transport chain. Your mitochondrion, which you remember is the powerhouse of your cell, has a bunch of enzymes bound to its membranes that work together in a pathway. So, this example, you can see the glucose is reacting, it's releasing its energy in the electrons, and they're going slowly down the steps. Each step is a redox reaction. They're releasing their energy nice and slow, and at the end, you get your product of water. So each of these enzymes, each of these little stairs in the diagram, does a different redox reaction and gives up or accepts electrons. So a lot of energy is released, and it's released slowly and in a controlled fashion so you can use it. Bioluminescence is another interesting energy transfer thingy. In bioluminescent organisms, light is actually emitted from metabolic reactions in different parts of their body, and this is called bioluminescence. The enzyme that makes bioluminescence possible is called luciferase, after the devil, Lucifer. Here's a picture of luciferase. It's this green guy. Luciferase adds a phosphate from ATP to the protein luciferin, which reacts with oxygen. The extra energy from this is given off as light. One animal that gives off bioluminescence is the firefly. I was very sad when I moved out to Oregon from Michigan that you guys don't have fireflies out here. If you ever have the privilege of visiting the Midwest or the East in the summer, you'll see these guys out on warm August nights in large numbers. And if you were a curious little tyke, you'll probably know that if you smush one of these, the glow from their butt will actually get on your hand, and the luciferase will keep catalyzing its reaction for a couple minutes until it wears off. On the left, you can see a group of petri dishes. The petri dishes have drawings on them, and these drawings are actually drawn in bacteria that are bioluminescent. This picture was on the cover of Nature magazine. This is one of the big journals of science, and it was done by a lady named Hunter O'Reilly. Now, anybody want to guess where Hunter got these bioluminescent bacteria? It was right from the abandoned Pacific Seafood in Charleston. There's actually a visiting researcher that comes out every year from Wisconsin and cultures bioluminescent bacteria from the guts of fish that they borrow from the fish plant in Charleston. And these bioluminescent bacteria are what were used to make this beautiful drawing.